Okay, so now for something completely different. Let's talk a bit about metabolomics. Um, so far, we've looked at proteins, peptides, and metabolites are very different from what we've seen so far in many ways. We discussed that in the first lecture already, that the size and complexity of the metabolome is largely unknown. Similar to the protein sequence databases, there are also metabolite databases that list all known metabolites, and these tend to be on the order of tens of thousands. So it's plausible to assume that the metabolome is of a similar size than the proteome, though we had this debate already how large that really is. But there are fundamental differences. Metabolites belong to a much wider range of chemical compound classes: lipids, sugars, amino acids. They are much more diverse than what you see on when it comes to peptides, and that also means that the technology we need for separation and identification is probably going to be very different, depending on which metabolite class we're really interested in. So you cannot do one separation to get lipids and amino acids at the same time. Proteins have a much more homogeneous chemistry in contrast, and that is why some of these tricks that we discussed earlier, like this averagine idea, work for peptides rather well, but for metabolites, this is tricky. Metabolites can have very complex structures that require a structural formula for a comprehensive description. So just writing down a sequence is also not going to work. And that will be one of the issues when we're talking about identification tomorrow morning. Metabolites are also on a different mass range. They tend to be much lighter. The average metabolite mass is probably between 100 and 300 Dalton, although they're heavier, some heavier metabolites. Proteins are heavier compared to that. They are on the order of 40,000 Dalton, which is why we actually fragment them into peptides for most applications. Metabolites comprise a heterogeneous set of biomolecules, or everything that is small in the system except salts and biopolymers, macromolecules. However, lipids and sugars are typically not directly included in metabolomics. They tend to be separate fields these days. We talk about lipidomics and glycomics because there are very special properties that come, or special problems that come with analyzing lipids and sugars. But if you go into these lists and see basically what are typical metabolites, in metabolomic studies, you find things like fructose 1,6-bisphosphate, ATP, glutamate, glutathione, basically all the things that you know from your standard biochemistry textbooks that are occurring in metabolic pathways at some point, all the cofactors, all of that. Similar to what we're doing in proteomics, you can also do metabolomics in two different flavors, namely targeted metabolomics, where you basically look for a well-defined subset of metabolites with high accuracy, and all of these metabolites are then identified. You might have heard of the Biocrates kit. That's basically a way to go only after well-defined, well-known metabolites. But of course, it is giving you only a very small glimpse into the true proteome. If we're covering 100, 200 metabolites, this is going to be hopefully interesting for some biological questions, but the vast majority of the metabolome will not be covered by these targeted approaches. That is why people are looking into non-targeted metabolomics, also called metabolic profiling, where we try to see as much of the metabolome as possible. Mind you, by choosing a specific analytical technique, you will always see only a certain excerpt of the metabolome. if you're looking for fatty acids, you will not see all the hydrophilic things. So you will never see the whole metabolome. And similarly, you will never see the whole proteome with one technique. Even if you see these metabolites, we need to define what C means. Right now, we're talking about detectability, but we're not talking about identification. And that's why we're doing it the other way around in metabolomics. We were talking about identifying proteins first and then trying to quantify them. Today, we sort of look at quantification to do this in a coherent fashion. And we will first talk about how to quantify these metabolites. Identifying is the harder problem in metabolomics, and that's what we will discuss tomorrow morning. In a way, everything we discussed this morning applies in similar fashion to non-targeted metabolomics. What are the differences? When we're looking at label-free proteomics, we're looking at a different mass range. That's a fundamental difference. What else is different? What is different on the technical side when you consider a non-targeted metabolomics approach and a label-free proteomics approach? Any ideas?
Vast majority of metabolites are only single charge.
The charges will differ. Metabolites are often only singly charged, that's correct. You might also want to look at both positive and negative mode. So, yes? Yeah, we have a different mass range, that's for sure.
You have to consider using multiple collision energies.
You need to consider different collision energies, yes. And also the gradient length is usually different. We tend to do proteomics on much longer gradients, several hours, and metabolomics experiments tend to be on much shorter gradients, 20 minutes, give or take a bit. So there are some differences, but fundamentally the technology and what the data looks like is very similar. So you could argue we can use the same overall workflow. We just do feature finding, map alignment, feature linking, and that is exactly what we do, but there are some subtle differences. The feature finding approaches are similar but not identical because isotopic patterns differ. We don't have an averagine. We have a very different mass range. And that means we need to use slightly different approaches to actually identify metabolite patterns. Other than that, you will see the same slides again. We start with a map, and in this map, we're now trying to find all the metabolites. So someone mentioned earlier that the metabolites tend to occur only in one charge state. But there's an additional complication in metabolomics that is not so frequent in proteomics. Say again? The adducts, exactly. So with metabolites, since the chemistry is so much diverse, there are many things that can happen. And one thing that really frequently happens is that other stuff sticks to the metabolite. So you see sodium adducts, potassium adducts, you see acetonitrile adducts. You see many weird things, and that can actually split the features. So again, even though you have only one charge state, you tend to see multiple features for the same metabolite. But fundamentally, it is still the same. We take a map, we look at the features, and we try to make sense of it. So we can recycle this. Yes, please.
Have you, in these proteomics workflows, specifically looked for adducts?
Yes. And you can do that. They occur in proteomics as well. They just tend to be less of an issue than in metabolomics. But you can look for adducts there as well. So we can basically recycle the same idea we used earlier today. We have a bunch of maps. In these maps, we do some feature finding, we do some feature linking and alignment, and that gives us a consensus map. I hope you've all seen that in the hands-on, really seen what that looks like. It's going to be very similar for metabolomics, to be honest. It's going to look exactly the same. You have to look very closely. If you look at the mass range, you can, of course, figure out what it is. But other than that, the data will look very similar. Now, instead of attaching a peptide sequence, we would try to attach a metabolite ID, a name, a database ID. And of course, again, we would like to see that there are different quantities attached to that. So when we look into feature finding algorithms, the first thing that we need to drop is this averagine hypothesis. Peptides have well-defined average composition, but metabolites don't. It's as simple as that. And that means you can have very funny isotope patterns occurring there. Another thing that we found over time is that these feature finding algorithms for metabolomics are even more sensitive when it comes to the algorithms, to the parameters. So tuning the parameters for feature finding algorithms can be a bit difficult. Different instruments, different metabolite classes, that all can lead to issues there. Another problem we found was sensitivity. We discussed that earlier today, and some people have asked already, how do you deal with noise, different noise levels? And I'll show you how we actually do that under the hood of the metabolomics feature finder, where we will go a bit deeper into the current algorithm. Why is sensitivity an issue? Sensitivity is an issue in particular in large-scale studies, because if you lose these features across maps, then you also lose comparability between maps. You simply have very sparse data matrices, and that can be a bit painful. So let me start with a short description of one of the feature finding algorithms, that is XCMS. That's actually a bioconductor package written in R. So this is not an OpenMS algorithm, but it serves to illustrate how feature finding can be done as well. The key idea in XCMS is similar. We extract mass traces, but here we do it by binning with respect to distinct masses. And then you detect peaks in these mass traces. By peaks, we now mean chromatographic peaks. And then align these mass traces in the retention time dimension using nonlinear dewarping. Mind you, what XCMS does here is actually not feature finding, not based on the definition that we used for features earlier. So XCMS basically does a peak finding on chromatograms. And there's some issues in particular when it comes to the binning, as we will see in a second. So how does this work? So XCMS starts with the raw LCMS data and then extracts ion chromatograms, applies a peak matched filter to find peaks in these chromatograms, then tries to align them across samples by defining global peak lists and create groups in dense regions, and then does some retention time correction. That's basically the alignment part that we do using nonlinear dewarping. And then fill in missing peak data. So go back into the data and try to find them. And then since you're in R already, you can just continue processing the data in R. Now there's some issues, and some of these issues arise from the way this is done, because binning, fixed width binning, causes some issues, as you can imagine. Because as the peaks sort of shift left and right, if the border of the bins are right around the mass, then the peak either lands in one bin or in the other. And that means it lands in one mass trace or the other. And you can see that here. These are two extracted ion chromatograms. And you see that this peak here is basically ripped apart. Part of the ion current lands in one chromatogram, part in the other. And XCMS has some tricks to combine these chromatograms to average that out, and then tries to identify these peaks here and apply some signal processing techniques to detect these peaks and reliably integrate them. Note that this does not yet do an aggregation of multiple mass traces. This is not done by XCMS, but there are external packages like Camera that can actually do that on the data sets. So XCMS has become a bit of a quasi-standard tool for LCMS-based metabolomics data, and recent versions include more sophisticated algorithmic details like wavelet-based peak detection, and that has been used frequently. We have a slightly different algorithm that is more aligned with what we do in proteomics, so full feature finding and a few tricks in order to avoid this binning that XCMS does, because we thought that causes more trouble than it's worth it. So how do we do that? When you look at such a feature, then we have the same problem that we discussed earlier. We want to find all the mass traces of metabolites. Oh, by the way, that is the figure I was looking for earlier, completely unrelated. Do you know what this gap here is?
Maybe, for example, in the urea sample, it will be the water region.
Say again?
For example, in the urea sample, yes, in the urine sample, that will be the water region, but it's deleted because it's of no information.
The water region, are you talking NMR? We're talking mass spec here, so no worries. It's the MS2. Yeah, this is where the MS2 is triggered, and you can already see this was not the ideal time point. But what you can clearly see is how this tandem, the collection of tandem spectra, actually causes gaps in the quantification. And if you trigger enough of these tandem spectra, then quantification becomes harder. All right, but what do we want to do? We want to find these mass traces, so we actually try to identify adjacent peaks in adjacent spectra that have the same or similar mass. And we then want to find this isotope pattern, so we want to assemble these mass traces back into one feature, and this feature would then again be represented in such a table where we had for every feature retention time, m/z, and intensity. And in this process, we would basically remove all these peaks that belong to this particular feature, end up with one feature. So the way this is done in OpenMS, and the tool is called Feature Finder Metabo, is we do a mass trace detection, and I'll delve into this algorithm in a second. And once we have mass traces that are in the same geometric region, we start asking, well, how can we assemble them? And before we do that, we actually check that they're contiguous mass traces. You remember this overlap, type 1 overlap. So we check whether we need to split these mass traces. In this case, actually, we have two features that are isobaric. So we need to split here. We would split these three mass traces off. And then we would assemble them. And by assemble, I mean we look for all possible combinations that would correspond to a metabolite. And the end result would be two features, one for this region and one for this region. So the heart of this algorithm, at the heart of this algorithm are three things: how to find mass traces, how to identify an isotope pattern, and how to score the feature. And I'll show you how that works. Let's start with the mass trace detection. A mass spectrometric peak is, for computer scientists, just a triple. It has a time, it has a mass or mass to charge ratio, m, and it has an intensity. Yeah, that's all the data is, a bunch of peaks in this two-dimensional data set. And now the mass trace is just a list of peaks. So we are looking for an ordered list of peaks where we order them by retention time, so that if you look at them afterwards, you get a chromatogram. That's what we call a mass trace. And the idea in this feature finder metabo, or in this mass trace finder, you can also run the mass trace detection on its own. It's a distinct tool for that. Is we try to estimate the local M over Z error. Now, what you see here is one mass trace that I've magnified. And looking at that, this is a bunch of peaks. And this is retention time. So the maximum of this feature would be around here, or of this mass trace. Now, when you look at that, some of these dots, some of these peaks are scattered a bit more. Why is that? And why are the peaks at both ends of the mass trace scattering further than in the middle? Well, you need to look at the intensities. Yeah, this here is where the intensity is highest, and thus the reproducibility, the reliability of the peak, the mass spectrometric peak is highest here. And as you go down, it becomes harder to actually call the peak in the mass spectrum. So the peak starts wobbling around, and eventually you will just find noise peaks around here. So the idea in this mass trace detection was the local error here is much smaller than the nominal instrument error, because the instrument error is averaged over the whole spectrum. But when you look closely, you can see that these peaks here are pretty close to each other. And what we're doing is a so-called online Gaussian density estimation. So we estimate the error based on the data. And we start by considering the nominal instrument resolution, or the mass accuracy of the instrument. That's the only parameter we need at this point. And we start with the highest peak here, again, the same seeding idea we discussed earlier. We are looking at this and ask, well, what are adjacent peaks that would be within the error bounds that we currently have? Now, this peak is adjacent. It's within the error. So we would add that to our peak list. Now, what happens is we can now re-estimate the error. And the error is going to be smaller because we didn't see the full error, the full resolution that our instrument could have. But we actually see that locally, these peaks are much closer to each other. So we can update this estimated error. And that's a very simple iterative update equation there. We can increase that and check for other peaks. And as we include more and more peaks into this, our error actually goes down. Because all the peaks are more or less aligned. And this is an error that is well below 0.1 ppm for most Orbitrap instruments, because the local error is much smaller. And now we simply stop as soon as we have peaks that don't fit the local error bound. And as soon as we are running out of peaks that are within these narrowed error bounds, we just abort the search. And this is a very nice approach, because it also gives us a local estimate of the mass error. And that is much lower than the tolerances we would need to consider otherwise. And that helps to correctly assign the other mass traces afterwards. Once we have collected this mass trace, we look at the mass trace ordered by retention time. So we look at the chromatogram. And now we don't have the problem that XCMS has with the binning, because we have collected all the peaks that are within the error bound, and there was no binning involved at any time. So that makes it a lot easier. And then you get chromatograms like this. And we would check that for peaks again and split at an appropriate position and identify already the full width half max of these peaks. Note that we are not assuming that there is an illusion profile underneath. Actually, the peaks, the chromatographic peaks in metabolomics look on average worse than for proteomics. And that's not surprising because, again, we have a much more diverse chemistry. So there are other interactions possible that you would not expect, and that sort of mess up the chromatography there. So once we have these mass traces and have them split up, we try to assemble them again. So in an ideal world, it would be straightforward to know that this monoisotopic +1 +2 traces are from the same metabolite. What do we know about them? Well, the first thing we know about them is that they have to coelute. They strictly have to coelute because they are coming off the column at the same time. So you would hope that you have a maximum correlation between these isotope profiles. But you can imagine as you go to very low intensity mass traces, this correlation can also become poor. The second thing you know is that the distances between these mass traces are not arbitrary. They are based on theoretical m/z distances. So you can figure out this is roughly 1 Dalton times the mass trace, the k over z. Now it's useful that we have this locally estimated mass error because we can simply assume there's a normal distribution underlying, and we can just check whether the distances would fit based on the locally estimated mass errors. We just ask, given these two mass traces, would the distance actually be correct? Would we be within the error bounds of the mass traces? And that gives rise to a pairwise scoring function where we simply assess, does mass trace A fit to mass trace B? That is the mass-based scoring. Then we have the retention time scoring where we're asking, are they really coeluting? And that is done rather easily. We're looking at the max and we're assuming there's also normal distribution between the shifts. And if they're shifted by too much, then we assume they're probably not coeluting. And sort of the trickiest part was this part here. How do we recognize? Oh, yes, please.
How do you decide what those reference mass traces? Is it based on intensity? Like, does anything that you're comparing to the reference have to be less intense?
Let me come to that. Yeah, we test again. We enumerate all possible combinations of or reasonable combinations of these mass traces, and then we do a scoring. And this scoring does indeed include the intensities. Nobody really had an averagine model for metabolites. So what we did is we simply trained a machine learning classifier and taught it to recognize these patterns, no matter what they look like. They are very diverse, but they're not arbitrary. So there are different classes of patterns, and we did that in a hopefully unbiased way by simply taking 24 million compositions. So all the small molecule space that we could lay hands on, subsampled that, and then trained a support vector machine to basically recognize these intensity ratios. And while there are different options for these intensity ratios to look like a metabolite, the support vector machine will basically recognize, yes, this looks like a metabolite because it has been trained on a very large data set. And then you combine these three scorings. You basically look at the intensity, you look at the coelution, and you look at the correct spacing of these guys. And then for all these hypotheses that you test, you pick the one that is high scoring. So how well does this work? It is actually not that straightforward to assess the quality of a feature finding algorithm. Can you imagine why that might be tricky? What's the reference? So that's a question concerning ground truth. What do you think if you would run triple distilled water on a reasonable instrument? How many features would you find? So there shouldn't be anything in there, right? But of course, you find several thousands of features. And that is because mass spectrometry is very sensitive. Some of that clearly might come from the column. Some of that might come from the tube that you had your sample in. Some of that might come from the autosampler in between. And you simply don't know. So we cannot say we just pipette together 50 metabolites and then we should see 50 features. That's not how it works. Also, if you buy supra-pure chemicals, then you also have all sorts of side products or degradation products in there. So there's no ground truth. So you can't just say, well, if we put in 50 compounds, if we find 50 compounds, then the feature finder is good. So what else could you do? Well, you could do a gold standard. You could just say, I measure the sample and then manually I pick these features. And if the feature finder finds everything that I find, then the feature finder is as good as I am. We found that there are differences in the quality of human annotators as well, as you can imagine. And also it's a pain in the butt to annotate that manually. And I failed to find computer scientists who have the patience to do that. So that's difficult. The other thing that you might argue is, well, if it's about quantification, what I really want to know is how accurately can I reproduce ratios? And all of these metrics come together, and ideally you would assess quantification algorithms on many of these metrics. They should be sensitive. What they pick up should be a true feature. They should be linear over a large dynamic range. So we did, for the assessment of this feature finder that was published in Molecular Cellular Proteomics in 2014, we did different things. Because the reviewers would never have been happy with purely synthetic data, we actually generated experimental data. So one thing that you can do is you can spike into a complex matrix small molecules that shouldn't be there, drugs, reserpine, terfenadine here, and you can check whether you actually see a more or less linear behavior. So we did that for a bunch of concentrations, and what you get is a correlation between the stock concentration and the feature intensity, and that looks okay. You can argue that R squared is probably not the best measure to also capture what's going on down here. You see there's also a little bit of a bump in there, but it gives you a good feeling if you see 0.997, that's not so bad over 3 orders of magnitude. I think that is not too shabby. The other thing that you can try is you can compare it to other tools. You can see how many features do you pick up. It doesn't give you like an absolute answer to your question, but it helps you to compare different methods. So what we did then is on this experimental sample, we also compared it to XCMS and camera, which does the feature assembly on the XCMS result. And you've seen this type of plot already. This is our bathtub plot again. In this case, we had 33 samples. And the gray bars are the OpenMS tool. The black bars are XCMS and camera. And we binned that a bit, this histogram. So these are the features that were found consistently in between 30 and 33 of the samples. These are the ones that were found in between 1 and 5 of these samples. And what you can see is we have twice as many features that we find consistently in more or less all of them. But we also have plenty of features that we find only in very few maps. So what is actually better? Which of these tools is performing better? So you could argue, well, OpenMS is clearly better. Of course, it has a higher bar here. It finds twice as many features with high reliability. Or if I like XCMS better, I would just argue, well, it finds 3 times as many features here. OpenMS finds 3 times as many features in the left-hand side of this figure. So this must all be crap. These are all singletons that are not reproducible. They are false positives. These are probably not features. So what's the answer? Yeah, better is hard to define. And fundamentally, we cannot answer this question on experimental data because we just don't know the ground truth. You can turn this around both ways. You could argue, well, these guys here might be interesting as well. And it's just that OpenMS is more sensitive. Since we don't have a ground truth, we cannot really distinguish sensitivity and specificity in this type of experiment. Based on experimental data alone, it's not easy or not possible to actually make that call. So one of the things we did is we actually wrote a simulator that simulates mass spec data. Because if you simulate the data, you know what's there or what should be there. So there's an LCMS simulator in OpenMS. And we went to a paper, so we went a larger metabolomics study where someone had gone to the trouble of putting all, it was manually annotated, all the compositions that they found in their sample into supplementary material. And we simply took all these compositions and synthesized a data set. This then takes the composition that's enough to predict the isotope patterns. And we added isotope profiles by predicted retention times. And then we added noise, various degrees of noise. Because that's a nice thing in simulated data. You can turn each of these knobs independently. That is hard in an experiment. And we can just check what's going on. And now we can actually determine recall and precision or sensitivity and specificity because we do have a ground truth. We can ask the question, how many of the features that we put into the simulated data can we pull out? And we can answer the question, how many of these that we pull out are really there? While this is not the same as the experimental data set, it gave us a pretty good feeling that we had a recall and a precision that was very close to 1. And that basically, to me at least, confirmed that many of these things that are on the left-hand side in this plot here are actually there. I mean, of course, we did more than that. We also went into the data and looked at it. But here with the numbers, you get the idea that, yes, sensitivity is higher and also specificity is higher. So you're actually getting what you would like to see. All right. Let's see how we can implement that in OpenMS. So we do some non-targeted metabolomics with OpenMS, and what we're showing you is a rather complex workflow. This is something that we did for a diabetes-related metabolomics study, and no, you don't have to read the names on all of these notes. I'm just going to give you some idea of how complex these analysis workflows can get, and then walk you through some selected aspects of that. So this was a diabetes study. We had 2 types of patients. We had metabolomics data, non-target metabolomics data. And we basically start here with this yellow part of the workflow that does the initial data processing, feature finding, and the rest is downstream processing. So this is where the quantification is done, identification is done based on accurate mass search. We will talk about that more tomorrow. And then ultimately we report these features and report their structures. And I'll walk you through some of the key parts of this workflow just to give you some idea of what that looks like. So the first part of that is the easiest part, and you should be able to understand that. This looks very similar to what you probably tried out for the label-free quantification workflow, except that we're now using this feature finder metabo. So we're taking all patients, the MZML files from all patients, we loop over each of these runs, and we find the features. Then we use the same algorithm, MapAligner post-clustering, because it doesn't care whether it aligns peptide features or metabolite features. At this point, it's just M over Z retention time. We map them, we link them, And then we basically have a table that we can push into the workflow. At this point, this has become a large data matrix that has rows for each of the features. And you can run that with a smaller sample set. You have to set just a bunch of parameters, noise threshold, then the estimated mass error. And if you're off like by a factor of 2, that does not really matter, because this adaptive estimation of the local error makes it comparatively robust to this parameter. But you should fundamentally know what to expect in terms of chromatographic peak width, and you should know what to expect in terms of mass accuracy. And these are things that everyone knows who is running the LCMS setup, so it's comparatively easy to parameterize this. And what it generates then at the end of this node, the output of this text exporter node is then a list that looks like this. We have a bunch of features: centroid RT, centroid M over Z, the charge of the feature, and here you can see we have usually singly or doubly charged features in this particular study. That depends on your conditions, depends on what you have in the sample. And then we have additional columns. These are the raw feature intensities without any normalization for each of the samples. Now we're using some of the table magic that NEU19 has. We want to massage this table into something that is easier to manage. And you can click this together depending on your study design, depending on what you actually want to extract. In this case, we are renaming the columns to make that more convenient. We are splitting some things up. We are doing something to these empty values. And that ends up in having controls and samples up here, so we actually know what's what. That's kind of useful with experimental design. Otherwise, nothing fancy. Then we do a statistical analysis and actually figure out what is differential between the two groups. We have to correct that for multiple hypothesis testing. This is all done in R. And as you can see here, we join the results of the identification and quantification, normalize intensities, that's variance stabilizing normalization. We do t-testing here. We correct for multiple hypothesis testing. All that is done in R. And what you see here in blue are attached sanity checks. We're trying to do the quality control as we go along in order to also figure out what's going wrong in these various steps. So we have plots like this attached where you can actually see, yes, this looks okay. We actually get something out that is meaningful. Or you have a quality control plot here for the log normalization. The box plot after normalization looks okay. So we would argue, yes, these samples are fine. The whole processing works. And that is something that I can really recommend to you. If you build these workflows, add in these visualization steps, make sure that you really know what's going on at every step, because weird stuff can happen at every step of your processing. And if you don't watch every single step, eventually you will lose control of your analysis. So go back, look at the data, and every single instance can be just one sample that is wrong and that throws off your whole analysis. So metabolite ID is another topic, one that we will discuss tomorrow morning in more detail. We can't do a sequence database search because there are no sequence database for metabolites. So there are a couple of things that you can do. You can search against reference spectrum databases. Basically ask the question, if I have this experimental spectrum, has anybody else seen a spectrum that looks like that before? That's what's happening here. We have a spectral matcher, and it basically gives you back results of identifications based on similar to a known spectrum. And the other thing you can do is, what are potential structures that have a similar mass? That's accurate mass search. Accurate mass, you remember the terms. Accurate mass is your experimentally determined mass. So you compare the accurate mass that you measured, that you get out of the feature finder, to the exact masses of compounds in structural databases. And if you have a certain threshold, certain tolerance, you can say, yes, this might be a candidate. You don't have a confirmed identification, but you don't have that in proteomics database search either. All that you have is a good candidate. And depending on which mass window you have, depending on tolerance, that can be like 2 candidates or it can be 150. And what this simple workflow does is it tries both methods. It looks at spectra from a database, but it also looks at a structural database and tries to find match just by accurate mass. And then this joiner brings it together. And there's one nice trick here. NEU actually started as a tool for cheminformatics for pharmaceutical industry. So really at the heart of NEU is already the functionality to draw nice structures. We're doing that with RDKit here. We're basically constructing the molecular structures and put that into an interactive table. And what you get out of this particular workflow looks like this. You have the structures here. You can scroll down. These are all the hypotheses that have been generated based on your metabolomics data set. And that I think is kind of cool because it gives you a direct handle on potential structures. You still have to go through them and decide which of these are right. But in this particular study, when we did the differential analysis, there were only 17 features that were really differential. By subtracting Plancks, by doing multiple hypothesis testing, usually you don't find that much. And then you can really look at these structures and see what makes sense, what are structures that are an option, and ultimately you will have to go back to the lab and confirm that if you really care about biomarkers anyhow. All right, with that I'm at the end, and I'll explain this identification part in more detail tomorrow